Hello, my name is Brooke Buckner, and this is my Unit 4 presentation over the article Gene Therapy Coming of Age. So the author begins this article by talking about how gene therapy has essentially been applied in clinical trials for over 30 years. However, methods of using gene therapy have resulted in some severe consequences, unfortunately, to the patients. And this could be the patients simply having a negative immune response to the trial, or the trial of gene therapy simply just wasn't effective. So in this article, the author looks to kind of go over the history of gene therapy and then basically what researchers have done over the years to make this more effective and ultimately safe for patients. In the early 2000s, scientists began making their own vectors used for gene therapy. And although this did have some success in modifying target tissues, this success didn't last long simply because the patient's immune response was activated by this foreign vector, and it didn't have a lot of success because of this immune response. So it really caused them to reject this type of therapy. However, the author then goes back to review back from the 80s and 90s how the idea of using retroviruses, which did have some success. Scientists then were able to effectively use or show efficient gene transfer of hematopoietic stem cells into these retro-C virus, and they were able to actually target T lymphocytes on a immune cell, therefore reducing the immune response. However, they also used two different viruses, including the lentivirus and the spumavirus. And the lentivirus was introduced because it could involve gene transfer in non-dividing cells, and it could carry larger, more complex cassettes of genes. The spumavirus, both actually the spumavirus and the lentivirus were preferred because they could integrate themselves into the coding regions of genes. Scientists also used adeno-associated viral vectors, or AAV vectors, and this was also successful because it replaced coding sequences with genes. However, there was a limitation to using AAV vectors because AAV vectors could only hold so many base pairs. It couldn't hold a large number of cassettes. So it could also, using AAV vectors, they found in studies with mice, they were able to transduce target tissues like the tissues in the cardiovascular system, tissues in the central nervous system, and so on. But unfortunately, patients still had a negative immune response to this vector. The author then goes to highlight the certain aspects of genome editing and how that could successfully be used in gene transfer. So one of the methods is that they could alter target DNA in gene therapy. They could alter target DNA by creating a double strand break leading to recombination of that DNA. And in 2012, the CRISPR-Cas9 was used, and this involved using a CRISPR-Cas9 nuclease that was programmed at the DNA site of interest. And what happened eventually was that it created what was called short guiding RNA that could target specific tissues that they were interested in. And by actually focusing more on the tissues that they were interested in, it could be more regulated and therefore lead to the desired gene expression. However, methods of genome editing, such as using the CRISPR-Cas9 nucleases, it's still in the very early developmental stages, and they really do need more safety data before it is implemented regularly among patients. Scientists then started focusing on more in vivo methods, really targeting the tissues directly and in implementing gene therapy directly to that target tissue. So there are some benefits to using this method as opposed to creating a vector outside of the cell by going inside the target cells or target tissues. This doesn't require any transplantation, any culturing, and it saves time because scientists don't have to collect cells. A study done in mice showed that using the AAV vector in the liver cells improved hemophilia. However, unfortunately, these effects were short-lived and the gene therapy really didn't work. It didn't last that long. And unfortunately, these animals had a negative immune response to the AAV vector, the capsid, or the outside of that vector. So scientists then began looking at a method called using chimeric antigen receptors, or CAR receptors, and this basically makes it so that the cell can recognize proteins when they are transduced into T cells, and hopefully, because these antigens are specific for specific T cells, and therefore hopefully limit or eventually take out that negative immune response. So the author concludes the article by basically saying that gene therapies, there have been some developments in the last 20 plus years since this started over 30 years ago, but it is still very much in the works, and still needs to be further fine-tuned before able to safely implement certain methods of gene therapy for patients. It is an organization called the National Institute for Standards and Technology. They have really worked on developing an assay or a method, or really a system showing that whatever method is chosen, it's supposed to be sent through that, and just to make sure that the gene therapy is safe, but also effective in clinical trials for patients. I think this article was a great indicator showing the progress of gene therapy, and I think it shows that ultimately when patients are really sick and have something really serious such as cancer, and they are maybe limited on their treatment options, gene therapy is something that they could look at. However, just with all other treatment options, especially with cancers such as chemotherapy, radiation, the benefits would most definitely have to outweigh the risk, and it would be on the provider really to discuss with the family of a patient and talk about what that could mean for their quality of life.